So as you can see here, I drew the semi-conservative model. So it's getting half of the parent strand, but it's getting half of the new strand also. And so we have anti-parallel strands. So that means that the pink and the black are going to be opposite. So if there's a 3' for the pink, there's going to be a 5' for the black. And you can see 5' for the pink, 3' for the black. So that's what forms the helix structure, that wavy structure, because they're running in opposite directions. So if we zoom in here, we see the bases, and so a thymine always binds to an adenine, a guanine binds to a cytosine, right? So we can see that. And so basically, what we have here is the phosphate groups. We have a sugar base, 5-carbon sugar, and we have a nitrogenous base. These things, I believe all of these things are called nitrogenous bases. So, and then, so those nitrogenous bases, if you zoom into here, you will see that there is an H bond between the T and the A. Wait, I think there was something that said like the T and the A have this many bonds, but then the G and the C have like 3 hydrogen bonds. But I don't know if I have to know that, so 3 here and this many here. So, and also these are nucleotides, and they're joined together here by what is called a phosphodiester bond or a covalent bond. It's covalent. But here, as I mentioned earlier, between the bases, the nitrogenous bases, it's just hydrogen bonds. So now I'm going to draw this black parent DNA in the pink color. And this is the new DNA that's following the parent DNA. And its nucleotides are, so in between here we have, like, for example, if we zoom in, we still have the A to T, G to C, we have that going on. So that's how this new DNA is following the parent DNA. And so this helicase is helping to unwind this way, right? And it's a bubble. This is a bubble shape. And actually, it's many bubbles, because this is what it would look like. But I'm just focusing on one bubble right now. And the topoisomerase is relieving the stress from the unwinding. And, okay, so there's like a bunch of winded up DNA here. Just pretend that's like, pretend this is the parent strand, pretend this is pink. So the topoisomerase relieving that stress, and, yeah. And also, there would be these things that are helping to keep the, DNA open, so that the parent DNA is open, so that we can keep making this new DNA. So these are called SSBPs. They're on both sides. And so if we zoom in, see this is RNA primer. And another one over here. So essentially what the RNA primer does is this serves as the binding site for DNA polymerase to initiate the DNA replication. So it would start here, right? It goes 5 to 3, so it starts 5 and goes to 3. So that would mean this is going this way, right? And this one has to be going this way. In other words, we can only add to the 3' end. Right, if it's going 5 to 3, then we can only add to the 3' end. Okay, so now I'm going to draw the DNA polymerase in yellow. I think this is 1. So here, and here, it's on the 3 end. Because as I just mentioned, we can only grow on the 3 end, since we grow 5 to 3. So the polymerase acts as the site of the growing chain, and it increases the DNA one subunit at a time. So a subunit would be the adenine, thymine, guanine, cytosine. So this is increasing an adenine, a thymine, whatever, increasing by one subunit at a time. Sorry, I don't know why it got blurry.
Okay, so actually what you need to know is this is the lagging strand, and this is the leading strand. And DNA polymerase 3 is synthesizing the DNA, the new DNA, on the lagging strand. Then DNA polymerase 1 is adding the parts on the leading strand, which is this strand. Oh, and something I forgot to mention earlier is this part, this direction where we're growing. It is the replication fork. Replication fork, where we're opening this way. That means this is the replication fork. Okay. Oh, and I forgot to mention something very important. So this RNA primer, actually both of them, they're synthesized by this like little enzyme called primase. So that's synthesizing this RNA primer. Same thing over here. There is a primase enzyme synthesizing the RNA primer. Primase.
So this is an analogy that Miss Kim gave me for why there's a leading and lagging strand. So this is the end of the road. Like, there's nothing, you can't build here. So no building. So we have someone who's just, so the construction is going this way, and this guy, he's building that way, the way construction is going, but you have this weird guy who's trying to, he wants to build this way for some reason, which doesn't make sense, because it's end here. So what he has to do if he wants to build this way is he has to go up and then come back. He has to go up, come back, go up, come back. And then each of these areas is the RNA primer. So, I mean, I don't 100% get it, but yeah. So essentially, we're just gonna have like a bunch of these like weird fragmented areas. And that leads us to having what we call Okazaki fragment.
So here's what it looks like in terms of my replication model. So in this first step, we have this RNA primase enzyme, and it's laying down a primer. And then the second step, we have a DNA polymerase, and he's extending this primer by adding these successive nucleotides one at a time until it reaches to here. And then a different DNA polymerase is going to get rid of this 5 cap from this other Okazaki fragment. And then this DNA ligase is going to come in, and he's going to be like the glue, and he's gonna glue these two Okazaki fragments together. And all of these steps occur on the lagging strand, not the leading strand, just the lagging strand because of its fragments. Also, another function of the DNA polymerase is that it proofreads, because occasionally you'll get an incorrect nucleotide, and then the correct nucleotide will replace the incorrect one.
So as I mentioned earlier, these have covalent phosphodiester linkages, but there is this enzyme called nuclease that can cleave these linkages and break this backbone apart, which is bad.
And something I forgot earlier is, in addition to this DNA polymerase, we also need a telomerase to add to both of the 3' ends. The telomerase is really important, and the polymerase can't add on its own. The telomerase aids in the replication and prevents chromosome degradation.